Monday. We're going to be going over neurodegeneration as a big picture. Kyle started saying it's like we're kind of telling a story. We're kind of going to need to blow through this because I think there's going to be some questions at the end and we can kind of clean it up at the end as well. I'm going to kind of introduce it. Kyle's going to do more of like connections and stuff towards the end. And the big thing is, understanding what a cell needs. We need three things: glucose, oxygen, and activation. So when these things are present, life can happen. And we're looking for life in these cells, right? Not death. We've also got, when it gets down to the nitty-gritty in terms of cells and biochemistry and all that, we're looking at a couple of receptors, because we're receptor-based organisms. Our brain and central axis thrives off receptor-based stimulation. So we've got a couple of receptors here. And if you guys have any questions, you can probably talk to Josh about this in the lower quarters. We've got what's called an AMPA receptor and an NMDA receptor. Now just kind of write this down. You can go into this further yourself if you'd like. For the most part, the AMPA receptor is responsible for sodium and potassium in and out of the cell. So with activation of that, you get changes in that, which also we know is the action potential, right? So this equals action potential. The NMDA receptor is more associated with plasticity and development of the cell, okay? More so genetic expression, things like that, okay? The NMDA receptor is more associated with calcium influx. Okay, man, you got me saying K now. It's driving me crazy. So the big thing with this is the NMDA receptor, so plasticity and development and genetic expression, is dependent upon AMPA receptors. So with these firing off at high frequencies, the AMPA receptor has to fire to activate the NMDA receptor. As a big picture, it's so we can get action potentials and so we can get genetic expression, synaptogenesis, health, life, things like that, right? Well, the cool thing about this AMPA receptor is that it's triggered by environmental stimulation or activation. So we all know what that is, right? Movement, anything, light, sound, all that good stuff. So that's all coming from the periphery. So in a big picture, we've got action potentials and plasticity is dependent upon movement. Does that make sense? And as chiropractors, this is a great way to, we have such a big effect on this simply because of this, right? So we've got the activation part here. We've got glucose and activation, looking at aerobic respiration. We all know that. So coming into the NMDA receptor, there's a couple of things you're going to need to know about this. There is a magnesium molecule blocking the influx of calcium for the most part. So this guy is going to be right here, kind of hanging out, until this AMPA receptor is activated, or until there's net phosphorylation in the cell. Kyle's kind of going to go more into that in terms of the death pathway. So what happens so far? We've got activation here of the AMPA cell, the AMPA receptor activates the NMDA receptor. This little magnesium plug is going to come off, let calcium in. You're going to get secondary messengers, so you're going to get your cyclic AMP, your protein kinase A, your mitogen protein kinase, and what else? Calmodulin. So those you guys know from cell phys. And then next, what that does is that creates basically the protein or the transcription type stuff that's going to happen in the cell, in the nucleus. And this is going to create life, all sorts of things for mitochondria. So it's going to create, let me start over. When this happens, you create genes and proteins. You transcribe these healthy proteins. And so these proteins are made for certain things. Can I go to that? These proteins will create things like neurofibrils, neurofilaments. So we're talking about structures of the cell. It's going to create more things for ATP. So in essence, we're starting to build this cell that is a little healthier. In addition, we've got, let's see, call it architecture. Proteins in general are negative, so they're more anions. So they are responsible for the negativity in the cell, so keeping that resting membrane potential around -65, give or take. Does that make sense? So there's some more things involved with that. But again, I'm going to let Kyle kind of finish that up. So as of now, let's go application to this kind of stuff. With glucose, what kind of problems do we see out there? We've got diabetes, metabolic syndrome, hypoglycemia, things like that. So diabetes, hypoglycemia, oxygen. Anybody? Rib subluxations, right? We deal with that every day. Smokers, anemias. And then activation. We're talking movement in general. People stop moving, they stop activating. You get a little less AMPA activation. You have less activation of the NMDA cell. You have less proteins made. You kind of get some stuff going on. So I'm going to let Kyle talk a little more about the activation, AMPA, and kind of what happens once all this takes place in the cell. It's going to go off and create other cool things.
Yeah, well, so it's like you said, you start with nervous stimulus. It's going to fire off the AMPA receptor. The AMPA receptor is going to then activate the NMDA, causing this little magnesium plug to dislodge. Okay, it's going to allow for calcium to influx and create protein. So that protein, like you said, is used for mitochondria. It's used for the architecture. It's used to create a better threshold within the cell. It's going to also create, so let's just write this down. So energy, it's understood. So increase absorption. We're also going to have what's called long-term potentiation. Has he gone over long-term potentiation with you guys? Okay, so all that means is that you're creating a nerve environment, presynaptically and post, that is more efficient. So it's going to reinforce all the structures. It's going to add new plasticity, which he pointed out that the NMDA is responsible for. So you're creating new synapses. You're also sending signals back to the other nerve cells prior to this to make sure that they create new synapses toward this nerve to make it more efficient. Okay, so everything is built around making the cell healthier, stronger, and more efficient. Okay, so that's the life pathway in a nutshell. You have all these three substrates available to the cell. The cell's able to run properly and able to become better. Now, when you take out one of these variables and you bring up, or something like, let's say diabetes happens, like Russ is pointing out, or anemia, you take out some of these substrates available to the cell. So now, when that happens, the cell loses the ability to run on aerobic respiration, and it's going to move into anaerobic, which I don't remember. Okay, so it's going to move it into anaerobic state, and most of you probably know that's not the most efficient way to create energy. And when you have that running, you're going to build up lactic acid within the cell, and that's going to do things like create. Let's write these down here. So red will be the bad stuff. You create free radicals, and these are going to start to pull apart the cell from the inside. you are also going to not, so since this is happening, you're not getting AMPA activation like you're supposed to. You will not have the calcium influx, and the NMDA won't get activated. So you will not, in turn, create protein. Okay? So there's no transcription really happening for the time being, and you're going to lose this negative charge within the cell, which helps maintain the threshold at negative 55 to 75. And now you're going to move closer to threshold. So you're going to creep closer and closer to firing off the cell at random when it's not supposed to. It's inappropriate firing. What is that, entropic depolarization?
Yep.
Yeah. So it's going to fire off at random, and it's not going to have the energy and cell metabolites in order to metabolize that process. So what you're going to see is the protein's going to break down within the cell. It's not being recycled properly, so protein has a half-life of 6 to 10 days, roughly. Yeah. So protein has a half-life of 6 to 10 days. It's supposed to be recycled within the cell through this whole process. If this isn't happening, those proteins aren't being recycled. They're sitting within the cell. They're not being dumped. They are also eventually dying off, and you're not maintaining the cell structure. So what happens is the nucleus will start to shift out of place as well. So all the structures that are supposed to be holding all those little organelles inside the cell together are now shifting out of place, and the cell is starting to deform. You're going to have, how many of you remember what the sodium-potassium pump is? Okay, so what does that run off of? What do you need in order for this to work? ATP. So like we said, protein's gone down, so you're not creating viable mitochondria either. And that means that your energy production's gone down, so your ATP has also gone out. And now you're stuck with ADP and an extra phosphate. Okay? So what ends up happening is you're not doing this anymore. There's no sodium being pumped out, potassium back in. Again, the threshold's sitting really close. Any little stimulus will set it off. You're going to have an abundance of phosphate inside the cell. And what phosphate's really good at doing is breaking down the membrane. So you're going to start to eventually move toward lysing the cell. Okay? And this phosphate will also, like Russ said, go up to the NMDA and eventually bind to it and cause the calcium to influx. So this is another way, this is almost like a survivability mechanism that the cell has in place in order to try to survive. So it'll bind that, calcium will flood in, you'll start to create proteins now, but this is toward the later stage, but these proteins will be kind of abnormal and mutated, and potentially you've turned genes on that aren't supposed to, and you could start creating things like tangles. So these are just abnormal protein structures, tau proteins.
You guys ever heard of tau proteins?
Lewy bodies.
You guys heard of Lewy bodies? Parkinson's?
Okay. Very good tangle. So this is a synuclein. Okay. So you're going to have abnormal protein production, and again, the cell is moving closer and closer to death. It's lysing within the inside. Sodium's starting to build up because it's not being pumped out. Now you're getting a flood of water coming inside the cell and it's starting to swell. So everything's making it easier for this thing to die off. Once this thing does die, you're going to have all these substrates flood the system, and the big one that you want to really recognize is that glutamate gets secreted outside of the cell now. And it's going to be all over, and glutamate's very excitatory, and it's going to excite all the surrounding cells. And if those cells are unhealthy, those cells are going to go through the same process this one is, because they just don't have the metabolic capacity to handle this. So those cells die off, and it's going to keep creating a domino effect until you hit a cell that can handle the stimulus. Okay. So how was that?
So one quick thing. Nice job, Hal. One quick thing, in addition with this glutamate, we've got this NMDA receptor, which is activated by glutamate, glycine, and also polyamines. So there's a appropriate things that need to be in place for this to happen. So appropriate amounts of glutamate, appropriate amounts of glycine, things like that. But like he said, when these guys start just spewing out all this glutamate, it becomes excitotoxic, right? And so you get too much glutamate, this will fire off, or this little magnesium plug will dislodge, you'll fire off this NMDA receptor, allowing calcium in, and it just perpetuates this whole process. Does that make sense?
Will the exterior glutamate fire off cells associated around the area as well, exciting those cells?
Right. So when this happens, boom, glutamate kind of goes everywhere. If this is kind of along the same pathway of an unhealthy pathway, you might think that when this other cells in this area are kind of going through what might be considered TND, and as soon as too much glutamate lets more calcium in to those cells, it's going to kind of go through the same process of just kind of apoptosis, things like that. So,
But this is where you guys can kind of get the idea that once the neuron cell is moved down this path, and let's just say it is just from lack of stimulus, then that's perfect because you can provide that stimulus with no problem. But let's say it's caused by diabetes. It's a blood sugar problem. You're never going to fix this with just an adjustment unless you address this. Okay? You could potentially be making it worse. There's the chance that you're going to make it a little healthier, but you're never going to have the full effect you want, and you're never going to be able to rebuild those pathways the way you want. And it could be any one of these: anemia, like he's saying, a ribosome subluxation could have some effect, but smoking, toxicity has a huge effect on some of these really key modulatory neurons inside the brain and brainstem. Okay.
in addition to that, the activation, guys, exercise. Exercise is huge. I mean, sometimes you might just need to come in, adjust a little bit for somebody's cerebellum, that might be going through something like this. You kind of make it a little healthier. And you're like, just get out there and run, do some movement, do these exercises, do some spins, whatever it might be, so these people can live on their own without having to come see you 3 days a week. I mean, sure, we got to make some money, but it'd be nice if you can fix these people and they can go out in the world and function, and they're doing this. It's like, why are you doing that? Oh, well, my chiropractor told me to go do this, and it's helped me. It's enabled me to go run for the first time in 2 years. It's like we can change lives just by understanding. It does get simple, but this application of neurodegeneration within the cells in the brain and the muscle. So
it's huge. Now when you look at somebody and you see dysfunction, you might be able to pinpoint it all the way down to this level. Okay? And when you have that, then you're going to have the greatest effect because you know exactly where it's at. All the way down to the cellular level. That's pretty huge.
Okay. So that's pretty much it. Questions, please.
I realize it's a lot of stuff.
What's AMPA stand for?
It's long.
I knew you were going to ask that.
It's really long.
I can tell you what NMDA stands for.
Okay.
N-methyl deaspartate. Oh, what's aspartate?
So that's sweet.
Okay, so that's kind of, we know that that's aspartame, that kind of thing. It's sort of excitotoxicity.
That doesn't help people.
What's that?
So that doesn't help.
I'm just saying, that's kind of one of the reasons why people talk about that stuff.
But yeah, you can hit that though.
How come we, when you dive into cell physiology and cis phys and all this, you don't really hear about the AMPA and NMDA, it's actual cell receptors?
It's because it's going to be over a lot of people's heads.
Okay.
And there's a certain amount you need to know for boards.
Yeah.
So we're actually working with Dave to get him, kind of introducing us, or at least having us come in and tag team this, so you understand it from a chiropractic standpoint.
Yeah, this is the basis of your adjustment for within the cell.
Yeah.
So I think it'd be pretty huge if he taught that because it can all of a sudden become real to you, and then it's all of a sudden self-phys is important.